Thanks, Anne, and I appreciate the recognition and the ability to share some of our thoughts. I think when Tim was speaking at the start of this afternoon, the point was well taken, the difference between clinical research and clinical science. So I'm going to present here some of the work that's been funded for a long time by NHLBI. For the past 30 so years, we've been principal investigator of the NHLBI Pediatric Cardiomyopathy Registry. It's a learning collaborative, and off of that, we spin off R01s such as this, which we had for 10 years. And this is an example of clinical research. But keep in mind that having learning collaboratives, whether it's CTSIs working together, or in this case, a whole network around the country, has enabled us to then look at clinical science. And the results of this work, have enabled us to figure out through learning collaboratives for these children with these rare diseases called pediatric cardiomyopathy, there was no improvement in medical outcomes from the late 1940s until about 10 years ago. But by having some of this work get implemented through networks, really where you look at best practices and you look at best ways to implement, we've cut failure of medical management by 50%, and these kids are living better now. But this is a little bit of some of the work that led to this. So this is the paper we're talking about. The first author, esteemed colleague Stephanie Ware, senior geneticist in Indiana. Couldn't happen without her. This is our centers. We've had the administrative coordinating center here at UV. And some of that team includes Miriam Mestry, Danielle Doffman, Lydia Grenier, Bill Power, and others. So really pleased. So in terms of background, cardiomyopathies in children is a disease of the heart muscle structure and function. And also when you go through these, there's more words than I usually like, but I wanted to make this less heavy on genetics and probably more understandable to those who might be here. So if you know what heart failure is, I apologize if you don't. It's harder for the heart to pump blood to the body, may lead to heart failure and death. And these are rare diseases. There are about 1 in every 100,000 children, work we've done and others, much less common, but much more lethal in children than adults, where it's about 1 in every 500. And in fact, about 50% or more of children presenting with symptomatic cardiomyopathy, such as heart failure, they fail medical management within the first 2 years after being diagnosed, and resulting in a heart transplant or dying of cardiac death. So these are really very significant conditions. And 50 to 75% of children have no known cause. Yet this is what's covered up there. This is the leading cause of heart transplantation from 2 to 18 years of age all around the world. That's why the purpose of this study was there's a critical need to determine the genetic causes of pediatric cardiomyopathies, to tailor therapies and counsel accurately. And so this study, when we wrote it and got it funded, was to identify the genes that cause or the variants that are associated with and may modify cardiomyopathy in childhood and influence the cardiomyopathy course over time. Our hope is by doing that, we can improve disease prevention and surveillance, early management, and prognosis. And it's enabled us to determine that certain personalized therapies, when we found some of these mutations, and we've then tested in genotype positive, phenotype negative newborns, we've been able to delay or completely prevent the expression of disease using targeted therapies. So it really has made a difference. So the methods in this study, we performed gene exome sequencing in 528 pediatric patients with cardiomyopathy and their families from 14 of the most major pediatric academic centers over the last 10 years. Those centers are noted noted there, and as you can see from the cartoon on the right, we really had children that by being in this and getting sequenced, we were able to come up with a significant percentage with clinically important pathologic mutations. And in this slide, what we're able to see is a little bit of the characteristics of these children, and it's a little bit male skewed. There's different types of cardiomyopathy. These children had DCM dilated cardiomyopathy, HCM hypertrophic cardiomyopathy, LV non-compaction, and restrictive cardiomyopathies. There's a significant family history in a lot of these children, but oftentimes the child's the index case. So in terms of what did we find? In this first results slide, we found that 32% of children with cardiomyopathy had at least one genetic cause identified, but many mutations were found. When we started this study, very few children presenting with heart failure around the United States were ever investigated looking at a genetic cause. It just wasn't part of the workup. But many rare and damaging mutations are found in our study. And for 56% of these affected children, the genetic variants were known to be rare and damaging gene mutations in other settings. And it was highly significant that the children with cardiomyopathy had these mutations compared to children who did not. And our control populations were two very large data sets of similarly sequenced patients around the world. Our second set of results is not only are they more common, but these same titles, dilated cardiomyopathy, hypertrophic cardiomyopathy, restrictive cardiomyopathy, there's plenty of adult patients that have these same labels. But when we really looked at it, the children's cardiomyopathies may have very similar names to cardiomyopathies in adults, but they often have very different, we've shown this before, causes, different risk factors, different paths of progression, different outcomes. But now we've determined that they have very different gene mutations. So the treatments in children must be tailored to the root causes of their cardiomyopathy. And although there's some overlap with adult mutations, they're often quite different and at times unique. And that's shown a little bit here. If you look at the two columns on the right, the one second from the right are adults with dilated cardiomyopathy. The one on the far right are children with dilated cardiomyopathy. And for laminin mutations and titin mutations, 2 out of the 3 on the bottom, dramatically different presentations between adults and kids, with more common abnormalities in adults than kids, and others as well. The other thing is when we looked at the difference between infants and children when they present. So in the figure on the right, in the lower, the gray are infants that were diagnosed around these major academic centers in the United States. And those in the black were children who were diagnosed not during infancy, the first 12 months of life, but later during childhood. And what you can see is there's a real potential for gene mutation discovery in infantile cardiomyopathy than in older children. Why is that? Well, again, many of the mutations we're finding in older children are similar to those well described in adults. But for infants, we keep finding novel gene mutations. And I think those are important because when we pick up in infancy, it enables us to intervene early when there's less severe disease. So, what we found here was that 36% of all patients presented with cardiomyopathy during infancy, yet a genetic diagnosis was only found in 20%. In contrast to those diagnosed beyond the first year of life, where 40% had a genetic mutation. So again, we think there's a real opportunity. The fourth bit of results is, I think, very relevant to the conversation we had earlier this afternoon. We further found that, for pediatric patients presenting with cardiomyopathy, a patient's ancestry really matters. And here we didn't use the NIH classification because we were matching this against global whole exome sequencing databases, and I'll show you that data in a minute, perhaps. But when we look at this, one example is the heart damaging gene mutation MYBPC3, myosin binding protein, which is a relatively common cause of cardiomyopathy in children of European ancestry, but was 100% absent among children with cardiomyopathy of African American ancestry. So, and some of these differences are very significant. So in conclusion, we showed that CCM, childhood cardiomyopathies, are a heterogeneous set of genetic diseases correlating with clinical phenotype for risk stratification and hopefully improve management and therapy. In the example that I just was trying to go over, based on this study, we really probably can't treat a 1-week-old African-American baby girl with the same therapy proven in a 75-year-old white man, because the causes of the cardiomyopathy are likely different. And there's also race, gender, age factors to consider in planning treatment. So the implications of this is that this supports the use of genetic testing for all children with cardiomyopathy, suggests that this will have clinical value. As I mentioned, we've subsequently, by using these, been able to show with NHLBI-funded work that we've gotten a significant improvement in failure of medical management by targeting therapies specifically. So again, that's that difference between clinical research versus clinical science. How do you actually, in those 14 centers I showed you, which represents some of the leading cardiomyopathy centers in the United States, the difference between outcomes within that learning collaborative versus the rest of the world is just night and day different in the same era. So again, the science of learning how do you implement clinical research to improve clinical outcomes is critical. And just as an example, the other thing is it results in financial and emotional cost savings. So an example of that is based on the results of this study. Now, only asymptomatic family members of a child with cardiomyopathy with the same familial cardiomyopathy gene mutation may need to receive expensive serial cardiac surveillance studies to look for the development of cardiomyopathy, instead of all first-degree asymptomatic family members, which has been the standard of care. Emotionally and financially, that makes a difference. So with that, I'll stop and thank you and see if you have questions.
Question?